So like I said, when you say bioinformatics, it includes genomics and proteomics, right? First, genomics, it handles the DNA sequence, the gene structure sequence, and whatever the information related to the genes and genomes. That's what it deals with, okay? The study and size of your genome, maybe it could be a human genome or whatever other organism's genome you're dealing with. It talks about that and it deals with that. And the physical structure of the genes and genomes. Your genome may be made of with the chromosome, maybe a linear chromosome or circular chromosome, so you deal with that. And then sequence information, of course, sequence of the coding region, non-coding region, regulatory elements, and all those things. And so yeah, it's a lot of information we're talking about. Okay, so how do you handle this information and how do you utilize them? That's a big question. So use of DNA as a starting point for discovery. What it means is, the 1990s, when we worked on the human genome, a lot of scientists worked on that. They had a really blue dream that, yes, once they figure out the sequence of all the DNA, then they thought, oh, they can cure a lot of, disease, genetic disease and everything. But, the answer was not that, because it's not just the DNA sequence that's important. It's also, how DNA sequence is transcribed into RNA, as well as how those RNA, mRNA molecules, or just RNA molecules are modified into mRNA, and then how they're transported to cytoplasm where the ribosomes are located, and then how they are translated. All those things are important. So it's not just DNA. This information is directly used to make protein. There are a lot more steps that are important. Okay, so if you think about the number of genes that are found from your genome, they may say, oh, human genome has maybe 30,000 or 40,000 genes, and that number doesn't match up with the number of proteins we have inside of a cell. There may be millions of different proteins. So the genomic study doesn't reveal all the information that we need to maybe cure genetic disease and all those human problems. So there are a lot more important things beyond the genome information or genomics, of course. Okay, so yeah, the bottom line is, yes, the increasing quantity of DNA sequence data that represented the challenges. So that was the beginning of the genomics. So both birth of bioinformatics and the extension of computational biology, meaning using computers, especially supercomputers, you are now able to not only store this DNA sequence data, also you can use the data whenever you want to. For example, you can do the sequence similarity search. For example, by entering a piece of DNA sequence, you can see if that sequence has a matching gene in the human genome or other organism's genome. You can do the sequence search quickly. Okay. So bioinformatics, that represents interpretation of the meaning of the information contained in the sequence data. It could be DNA sequence data or protein sequence data, RNA sequence data. Okay. So it heavily relies on computation, the mathematics, as well as the computer science. Okay. So gene counting, functional genomics, structural genomics, medical genomics, plant genomics, evolutionary genomics, they are all part of this field. Okay, so how many genes are located in your genome? That's important, right? Functional genomics, what do those genes do? Okay, and structural genomics, how your chromosomes develop the structure. Okay, what is the histone structure? What is nucleogen structure? All those things are important because the structure should be changed when the gene is expressed. Of course, medical genomics, again, genetic disease, how they are caused, developed, what gene mutations are important for those kind of things, right? Plant genomics, plants are very important because a lot of them provides a lot of important benefits for us. So we need to know a lot about the plants. Evolutionary genomics, of course, when you study evolution, you can compare not only the structure, you can also compare DNA sequences. So these are all dealing with the vast amount of information. Okay, so when you deal with all those information, you need the computer science, of course, way to analyze information. So life science, so through your biological study, you can come up with the DNA sequence or RNA sequence or protein sequence, okay? And then the information science will help you to better store this information and then better analyze this information. So it's the fusion of those two together, which is the bioinformatics or genomics or proteomics, okay? So like I said, the proteomics, similar to genomics, but you're not dealing with DNA sequence. Instead, you're dealing with the protein structure and protein sequences, okay? So it provides comprehensive understanding of how the structures and functions of all the endpoint of gene product, which are the proteins, okay, in a specific type of cells are integrated. So meaning proteins are made. I mean, there are a vast amount of proteins that exist inside of a cell. For example, human cells may have millions of different proteins. Actually, there are more number of proteins than number of genes that are found from your genome. So how can you make millions of proteins with only 30,000 genes? That's a complicated story. Okay, there are a lot of modification processes that will allow you to make a lot more proteins, because proteins are very important to maintain your structure, develop your cells, and then carry out whatever the metabolism, cellular processes. They are all managed by protein molecules. Okay, so proteomics is very important as well. So genomics and bioinformatics are necessary but not sufficient for understanding cell behavior and pattern, because proteins do serve a lot of important functions. So without talking about protein, you have more than half of the information you need that's important for explaining what's happening inside the cell. Okay, so how proteins are expressed, under what condition, what proteins are made, because cells respond to the condition and signals, and they change their gene expression pattern dramatically. Okay, so that's very important. And then structure of all those proteins, which ones are expressed, and how do they maintain their structure, how do they get activated, how do they change their structures. That's important, right? Interaction, how do proteins interact with each other? We talked about, the assay method that will allow you to detect the protein to protein interactions, right? We talked about 4 different methods. Is two hybrid screening, FRAD is another, immunocore precipitation, GST pull down. So those assay method will allow you to understand in protein to protein interactions. All those informations are accumulated, and you need to be able to use those information to understand what's happening inside of a cell. So we're talking about a lot of information in the proteomics area. Okay, and then bioinformatics and its application, of course, is very important. Biological information includes widely used software for analyzing DNA, RNA, and protein sequences, as well as databases and type of searches that be performed on DNA, RNA, and protein structure functions. Okay, so type of searches meaning, if you are studying on one gene or one protein, okay, your question will be, can I find this gene from different organisms? Okay, do other organisms have similar proteins? And if they do have similar proteins, what do they do in those organisms? Okay, by comparing the proteins or DNA gene sequences with other organisms, you can better understand the structure and function of your DNA or RNA. Okay, so you should be able to handle the information that's already available to you by using the software database, analysis tools. Okay, for example, BLAST is a search function that will allow you to do a sequence comparison. Okay, if you are studying on a human gene, for example, you can maybe compare your human gene against maybe plant gene or animal gene or bacterial gene and see how similar they are or how different they are. Okay, and the Medline is on search engine again, it allows you to find all those professional journal articles. You can easily find them. Okay, so Antres also integrates DNA and protein sequence data with the associated abstracts and citations and journal articles together. So these are all important search engines. Whatever the DNA sequence you studied on or protein structure you studied on, they are all normally published. There should be associated papers, research articles that are out there. So if you use these search engines, you'll be able to find those information. Okay. So this diagram may be a little outdated. It's from 2001, about 19 years ago, but even back then there are so many available entities that were out there through World Wide Web, so BLAST search, sequence search, FASTA, and all these tools, as well as different research institute maintaining their different genomic databases and search engines of different kinds. So today there are more of entities out there where you can find more important information. Okay. So how to use the internet to look for information on DNA, for example. Okay, so that's very important. So if you are doing some kind of research, so on maybe DNA sequence, so GenBank is a very important place where all the genetic information, DNA sequence, has been deposited. So if you go there, you can identify a gene and its sequences easily. So GenBank is a really important resource for you. And then so nucleotide home, against NCBI. So if you click on it, there's that address, website address. So if you visit the NCBI website, it provides you all the resources all together. So let me go there right now. So I'm just doing the NCBI, maybe PubMed, Google search. Okay, so if you do that, you will see this PubMed, the website. If you click on it, so you go to there. Okay, so PubMed is a place. It is funded by the government, NIH, National Institute of Health. Okay, so if we do a keyword search here, if you type in whatever the keyword, you can do the literature search easily. So let's try to type in search word of something. Let's say bioinformatics, for example. And then ask it to search. So yeah, that's what you get. So it showed you so many papers. About how many papers? About 349,000 papers that had the word bioinformatics in its title or abstract. Okay, so it's allowing you to pull out all the published journal articles that has something to do with the bioinformatics. And if you want to narrow your search, you can add more keyword, maybe genomics, for example. So if you combine the words, you will see the number of papers is getting smaller, 197,000. Still, it's too many. And maybe you can do proteomics together. And so yeah, now it's 1,200 articles. So by adding more keyword, more and more, you will be able to pull out papers that are more relevant to your search. Okay, so that's a great search engine where you can do a lot of literature review that's helpful for your research. Okay, and besides this literature search tool, you can also do other searches. For example, let's go to this website, NIH National Library of Medicine. Okay, if you go to that home page, you will see all these function keys here on the left side. It has all these resources possible. So all resources, chemical and bioassay related resources, data and software related, DNA and RNA related resource, domains are structure related, gene and expression related. So all these search engines will allow you to do more searches on these topics. For example, DNA and RNA. Let's see what you can find if you click on it. So yeah, database about DNA, assembly, biosample, GenBank right there. Okay, influenza virus. Okay, so many informations available. Reference sequence, sequence read archive. Okay, database, for example, if you want to go there, assembly, bio collection, biosample database, download submission. Okay, so these are all really important resources. So quick link here, look at it, GenBank's sequence. If you want to study about proteins again, yeah, protein related resources again. Okay, database. Human protein interaction database, identical protein groups, and there are so many of those important kinds. Okay, Okay, so yeah, understand the importance of that database, NCBI. That's a really great resource for your research. Okay.